How's everybody doing out there? Dr. Gillard here for another lecture. This is Tuesday, week 5. This is the first of 2. It's the short lecture on Tuesday. It's week 5. It's spring 2020. It's spinal anatomy, and here we go. We started talking about the cartilaginous vertebral endplate last time. Or we talked about that before, but let's make it official now. Remember, this is the third component of the intervertebral disc. What are the other 2 components? Or you tell me, what are the 3 components of the intervertebral disc? Really easy question. Nucleus pulposus, annulus fibrosus, and the cartilaginous vertebral endplate. Now, different professors, different schools, different boards, you might just say vertebral endplate. I don't like to do that because there's a bony vertebral endplate and there's a cartilaginous vertebral endplate. So I like to keep them separate, but just watch out for that one. There's a little controversy about this, though. Bogduk says it's still kind of debatable. Some believe that the cartilaginous vertebral endplate actually is property of the vertebral body. But because it's really not stuck to the vertebral body well at all, I think it's safe to say, and most authors consider it part of the disc, not the vertebral body. Here it is. We've looked at this before, right? We have a kind of a toothpaste-like nucleus pulposus in the middle here. And then we have these lamellae. Remember we talked about the lamellae. There's about 15 of them, 20 to 50. Everybody's a little different. But the lamellae actually go into the vertebral endplate and completely corral the nucleus pulposus. So pretty cool, right? What else do I want to say about that? Notice here, though, things are a little different. And we got slides coming on that. But this is called the ring apophysis. And this is actually where the bone grows from. There is no endplate. There is no cartilaginous endplate there. In fact, the lamellae don't encircle anything there. They anchor right into the bone. And therefore, these lamellae get a different name in this region. Some call them Sharpie's fibers. Those are important. Sometimes pulling and tugging on those can grow what's called a traction bone spur that is lifted up a little instead of growing down here at the kind of the disc endplate junction. They grow up a little higher. You'll get that as you kind of move on up. Histologically, it's kind of a weird tissue, this vertebral endplate. It's cartilaginous vertebral endplate. It's made up of 2 layers of tissue. There's a fibrocartilage, which contacts the nucleus pulposus and interannulus fibrosus. And then there's a hyaline cartilage that's attached to the vertebral body. And the hyaline cartilage, where it attaches to the vertebral body, is not the greatest in the world. In fact, you can easily rip the disc away from the bony vertebral endplate. In fact, you can rip some cadaver studies. You can rip it right away, and the tear can go right into the disc itself. Not true for fibrocartilage. That is really strongly connected. Nucleus pulposus and interannulus are connected quite strongly together. That one can't be ripped apart. All right, we talked about Sharpie's fibers here. Oops, let me get my highlighter disappeared on me. Yeah, Sharpie's fibers are the lamellae that insert into the ring apophysis. I always ask that question. And they're strong, so this is actually considered the intervertebral ligament by some authors, right? Because really, technically, you're going bone to bone, so that would be a ligament. Okay, anyway, that's the deal with Sharpie's fibers there. They connect directly to the ring apophysis. The outer lamellae are called Sharpie's fibers. Very strong, type 1 collagen, super strong. And that would be the intervertebral ligament if you ever see that. It's a member just like the anterior longitudinal ligament, posterior longitudinal ligament. The intervertebral ligament is Sharpie's fibers. Ring apophysis, we talked about it, but on the superior and inferior surfaces of the bony endplates, there is a thin peripheral margin of bone called the ring apophysis. We looked at that. That is a site where secondary ossification centers, bone-making factories, grow. In fact, the proliferation of bone from the ring apophysis is what makes the human grow a little bit taller. The vertebral bodies grow a little bit taller in the vertical axis. So that's kind of their claim to fame. Here's a different view, but you can see it here from the top view. That is called the ring apophysis. Okay, top and bottom. So there's an osseous. We talked about this. There's an osseous top and an osseous bottom. Those are called the superior osseous vertebral endplate or the bony endplate. Right? And don't mix those up. Make sure that's why you should use bony endplate to distinguish between cartilaginous. I think we talked about that already. Vertebral endplates are actually quite thin. They're really 1 millimeter thick, so the lamellae really scrunch together going through there. Again, they're absent at the periphery where the ring apophysis is. Best to call these cartilaginous vertebral endplates. Kind of a review slide, I guess. They cover the superior and inferior part of the bony vertebral endplates, except they do not cover the ring apophysis. Common mistake is to think they cover the entire bony endplate. They don't. All right, now we're going to fall down a rabbit hole because we're talking about the nucleus pulposus and why it's 80% water. Well, the reason it is because it's made up of glycosaminoglycans who kind of conjugate into a structure called a proteoglycan. So we need to talk about this. This will be also covered in biochemistry, but it's my domain as well, so let's go through this. So gags, we'll just call them, they're the main molecule of the nucleus pulposus. There's also some in the annulus fibrosus and some in the cartilaginous endplate, but there's more, especially in the annulus fibrosus, there's more type 1 collagen there and less GAG. There's tons. The primary molecule of the nucleus is GAG. That's not true for the annulus fibrosus. So we need to dig into these a little bit. So another word, an old AKA, is mucopolysaccharides. That was, hopefully that still won't be on boards, but you never know. They are considered long, unbranched, heterogeneous polysaccharide chains. So that's a mouthful. Heterogeneous means they're made up of more than one, the multiple types of molecules. Polysaccharide, that's sugar, so they're made up of sugar, different types of sugar in a chain. The repeating units, more specifically, what type of sugar, they're made of disaccharides. We've heard that word, disaccharides, right? But you're thinking, maybe sucrose, right? But this is not the type of disaccharide that we're thinking about. You're thinking about these guys, right? There's sucrose molecule, right? That's a disaccharide. Lactose, maltose. But what's the difference between these and the gags we're going to look at is there's no polar groups here, right? Polar groups are super hydrophilic. They love to grab and bond to water. So our run-of-the-mill, kind of our GI disaccharides, if you will, they don't have carboxylic acid groups or amine groups or even sulfate groups on them. So these guys wouldn't do us any good if they're inside. So the key is the proteoglycan. That's the key of the disc. Proteoglycan is made up of two types of gags on a core protein. And here's SpongeBob. This is proteoglycan Bob because they're really like sponge. They absorb water like crazy. And it's not that they really absorb water. They bind water and hang on to water. And therefore, the substance gets real slippery and watery. Almost, it's a terrible example, but snot or mucus that you spit up, it's all slippery. That's got gags in it. The synovial fluid inside of knee joints that you've seen in the cadaver lab, maybe you haven't seen them this quarter, but it's slippery to the feel. Same thing with the pericardial cavity, very slippery. And around the lungs, super slippery. Those are all from gags that make it slippery like that. Why? Because gags are super duper hydrophilic. They're super polar, and they can bind water like crazy. They also repel one another, right? They're super negatively charged, and therefore they will stay their fair distance, like social distancing. It's like gag distancing. There's 6 major types of gags in the body. All of them are sulfonated. They contain at least a sulfur atom with an oxygen, except for the weird, strange, oddball hyaluronic acid, aka hyaluronan. Only 4 of the gags are found in the human disc, and let's take a look at them. 4 types of gags. I guess I can get rid of, let me make a note to get rid of 449. I kind of experimented with a new slide here. So there's the chondroitin brothers, chondroitin 6 sulfate, chondroitin 4 sulfate. Most of the time I think boards will probably just say chondroitin, but you never know. It's 4 and 6. There are other types of chondroitin. Keratin sulfate is the other one, and hyaluronic acid. This is the little oddball one here, but it is hanging around, as we'll see. We'll make use of it. But really, the main proteoglycan of the disc with functional gags that grab water are chondroitin sulfate and keratin sulfate. Hyaluronan is in there as well, but it serves a little different purpose. So again, the gags are special because they are super hydrophilic and they can attract water like crazy. Why? Because they have super polar groups or super hydrophilic groups, sulfates, carboxylic acids, and amines. Amines aren't the greatest for attracting water, not like sulfates and carboxylic acids are. Carboxylic acid, that's the C. You'll see this written all the time, right? It's got a C double bonded. Oxygen sticking up and a hydroxyl group on it. We've seen that, right? I haven't had biochem for a while, but anybody who really studies biochem knows the dreaded carboxylic acid group. Right? That's a carboxylic acid group, and that's just kind of on a sticking off the end of a chain there. That's a carboxylic acid group. I won't draw the sulfate group. I think that's getting too deep. I'm not going to ask you to draw that, but a lot of you know what that is. Well, therefore, these groups will hang on to water, as we said. They have a great water trapping ability, and they give that ground substance or the total substance of the disc a real slippery characteristic. The average disc is about 80% water, so the reason for that is all of these polar groups that are on there. Which are the best water trappers? The chondroitin 4 and 6 sulfates are really good water trappers because they have 2 hydrophilic groups. They have a carboxylic acid, or aka carboxyl group, and they have a sulfate group as well. Keratin sulfate, it does not have a carboxylic acid group. Therefore, it can only bind about half as much water. Right, so it just has a we'll see, it has a sulfur group on it. And and hyaluronic acid does not have a sulfate group on it. So keratin sulfate, no carboxylic acid, and hyaluronic acid doesn't have a sulfate group, so it only binds about half as much water. Let's take a look at the real specimens here. So here's a chondroitin 4 sulfate here. You can see it's got a carboxylic acid group, super polar, and it's also got a sulfate group here. Right? They all have these nitrogen amine groups on them, right? Every single one of them. They're not the greatest at binding water. It's these, it's carboxylic acid and sulfate groups. So we got double binding power here on a chondroitin 4 sulfate. Chondroitin 6 sulfate, almost the same setup, double binding, real good at grabbing water. But keratin sulfate, it's got a sulfate group, but that's it. And then a hyaluronic acid has a carboxylic acid group, and that's basically it. So these guys are 50% water binders, not the greatest. Hyaluronic acid's not. That's a weird molecule. It's another 50%er right there. Right? So some gags don't like to be alone either. They're lonely. They don't like to be, although they don't want to be super close together, they like to conjugate. So 3 of the gags, in fact, don't like to be alone. I call them the sociable gags, and that's just my word, so I remember them. And they're the keratin sulfate and the 2 chondroitins, chondroitin 6, chondroitin 4 sulfate. These guys like to be around each other. They like to socialize. And in order for them to socialize, they have to have a recreation center to socialize. No, of course, they don't go into a recreation center, but they do have to have a place to kind of hang out and meet. And that's where the core protein comes in. So that's really important. You actually name different types of gags via its core protein. It's one way to name them. It's also secreted by nucleus pulposus cells. And that is the place where they conjugate. So they bind, the 3 sociable gags bind on a core protein, and that's where they hang out. And when they bind together, just like if they're all hanging out inside here together, talking, playing tennis or playing ping pong or whatever they're doing in here, this whole molecule now gets a name. We don't call it the recreation center. We call it the proteoglycan. Okay, so the gags plus the core protein that they bind on are collectively called a proteoglycan. Got it? And this proteoglycan makes up the main molecule in the nucleus pulposus, and it's in the annulus fibrosus as well. There's 3 different, here's just a run-of-the-mill. Note the 3 different gags that we have on this core protein. So the black thing's the core protein. We have a chondroitin 6 phosphate hanging out there. We have a chondroitin 4 sulfate hanging out there. And we have this keratin sulfate hanging out there as well. And there's the core protein. Together, this whole setup, and we won't talk, there's linker proteins, and we'll talk a little more about these domains in a minute. But this is a proteoglycan. This is proteoglycan Bob. Right? Proteoglycans are found in 2 places in the body. Well, the disc is one more. That's what we're talking about. But they're in the extracellular matrix and ground substance of different types of body tissues. Cartilage in particular, all the joints, your elbow joint, your wrist, your finger joints, even the joints of von Luschka, even the sacroiliac joints we talked about. They all have a lot of these gags making them slippery inside. They're also seen on the outer surface of cell membranes. I won't worry about that right now. We said they're made of a protein core, and the gags are not polar bonds. They're covalently bonded to the gags, so it's very strong. And there's a lot of different types of proteoglycans. Some of them can have up to 100 gags bound to the core protein, so it can be a gigantic molecule. There's the core protein. We can have them bounded. I'm not going to draw 100 of them, but there's on each side there's one. So it starts to look like that, and that thing is real slippery if you get a whole bunch of those together. So gags are sociable, but they're not super sociable because they are negatively charged. And you know what happens from probably junior high science days. What happens when you push 2 magnets together, the north pole of 2 magnets together? There's a force. They repel each other. And the same thing happens when the gags are on their core protein. There's the core protein right here. And they are perfectly parallel to one another because they can't contact each other, and therefore they get this bottle brush appearance as the classic description. Notice how that proteoglycan looks kind of like that, doesn't it? Except for the end of it. So classic bottle brush appearance. Why is that? Because these are negatively charged, these gags, right? And this is a gag, one gag right here. This is one gag. And they're kind of too close together, but so each one of these, all these gags bind to a core protein, this black thing, and therefore you call it a proteoglycan. And the type of core protein it is names what type of proteoglycan it is. We're not going to get too deep. We'll talk a little bit about families. I guess we're going to do that right now. So the families of proteoglycans. So proteoglycans are grouped into different families based on what type of core protein, or specifically they're named from the gene that makes their core protein. All of GAGs have a core protein. They're not named by their core protein. They're named by the gene that makes the core protein. Okay, one important family member we have to talk about is the aggrecan family. It's a terrible name. Bogduke is still using it, though. Modern biochemists are calling this the lectican family. And you'll see in a minute it gets really confusing because one member of the aggrecan family is aggrecan. So it's like, what? It doesn't make sense. So it's better to call them the lectican. The Kramer uses the same aggrecan family. It's better to use lectican, but I don't know on medical student boards how that will be, or PT boards, but chiropractic boards, they're still calling it aggrecan family. So members of the aggrecan family are found in the disc and cartilage, and yeah. So let's meet the aggrecan members of the lectican or the aggrecan family of proteoglycans. There's several different types. One thing in common that all the different family members have is they have to have chondroitin sulfate attached somewhere. It can be 4, it can be 6, it can be a mixture of them, but they have to have chondroitin sulfate in there. They can have other and different types of GAGs, but they have to have chondroitin sulfate. The one on the disc has keratin sulfate as well, and that makes up most of the disc, but that's not true of all the synovial joints. But let's look at the family members, the aggrecan family members. So as I said, one of the most important members of the aggrecan family is aggrecan, the proteoglycan aggrecan. It's made by chondrocyte cells in cartilage. It's the most studied by far. It's made by the nucleus pulposus cells as well. And it's confusing because aggrecan is a member of the aggrecan family. That's why it's better to call it lectican. For example, who's another member of the aggrecan family? Versacan is another one. This one is made. It still has it still has GAGs attached to a protein core, but it's a little different. It's made by fibroblasts this time in smooth muscle, and so this one is found in blood vessels and dermis, places like that. It's not found in joints. Versacan. Another one called neurocan, and this is made by neurons in the CNS. There's another one made by astrocytes called brevican. Who are these? These are proteoglycans. They all have a core protein and different types of different types of GAGs bound to them, and they're all under the main category of aggrecan. These are all aggrecan class proteoglycans. Then we have one called the SLRPs, the small leucine-rich proteoglycan family or members. So these are proteoglycans as well, but they're also under the main category of aggrecan. Got it? We need to talk about these because they are important for the disc. There's two key ones that we want to talk about, decorin and bioglycan. And I'm not going to get into this any more than this. There's a lot of research on both of these, and if you have a mutation in the gene that makes these, it does have a role in the genesis or the formation of type 2 diabetes as well as obesity. That's all we'll say about that. In addition to the chondroitin sulfates, the SLRPs here, they have to contain dermatan sulfate. So these proteoglycans interact with certain growth factors that they run into inside the matrix of a disc. And why am I even mentioning them? Because they're really important for repairing the matrix of the disc if it gets damaged from maybe squatting over the weekend or playing football or something. Mutations in these genes do increase your risk of getting degenerative disc disease. Okay, now let's talk about the aggrecan, the proteoglycan called aggrecan. This, again, is a member of the aggrecan family, so I know that's confusing. You could say it's a member of the lectican family. It's not as confusing. But this is by far the most common type of proteoglycan that is found in the intervertebral disc. Okay, so aggrecan is super important. This is the type of structure that you see. It's made of keratin sulfate, and it's made of the two types of chondroitin sulfate. Okay, we kind of looked at, we saw a picture of it. The bottle brush is what back there is a picture of it. The structure, it has a core protein. A little more specific this time. It has 3 coiled regions, which are called the globular domains, 1, 2, and 3. Between these globular domains are some straight regions called extended domains, E1 and E2. And that's the extended domains is where the gags bind to and hook up. Okay, aggregans are not a gigantic molecule, considered moderate to large. There's about 70 gags on there. It can be much bigger than that. Here is a basic aggrecan molecule that you'll find floating around the disc. And it has keratin sulfate would be here, the shorter ones. And then the chondroitins would be on the E2 domain here. Did I write that? I don't think I wrote that in there. But yeah, that's the setup that we have here. We won't talk about the globular domains, what they do, but if you want to hook up with another molecule, there is one called an aggregate. Sometimes these can become members of an aggregate by binding to the G1 domain. These guys tend to prefer to be alone, though. They can be that member, but they cannot. We'll look at that right now. So aggregans are super hydrophilic, right, because they have chondroitin and keratin sulfate, which are double grabbers, right? So they bind water like crazy, and they coil up, and that's pretty much what you see here. Makes them really slippery. The negative charges kind of repel each other. And they're found in some other slippery substances like mucus, the intervertebral disc, synovial fluid, vitreous humor, pericardial, pleural, peritoneal. The 3P types of fluid are slippery. Make sure you grab that when we get back and you get in lab. Grab that pericardial sac, pericardial cavity. Put your fingers in there. Put your fingers around the lungs and feel how slippery the pleura is, the parietal pleura. You'll see that on the chest wall. It's super slippery because of these things. Okay, some of the proteoglycans do like to group together. So gags, they like to be sociable. They bind on a protein core and make a proteoglycan. But some of the proteoglycans like to be sociable as well. And one good place for them to meet up and hang out is that oddball hyaluronic acid. It's almost like a protein core in a way. Only it's not. The protein core is found in the protein core, of course, makes up the proteoglycan. I need to turn my phone off the things. Okay, so a good place for proteoglycans to meet and hang out is the hyaluronic acid molecule. Okay, specifically, they bind via a linker protein, which I showed you, and they can hook up, and they create this gigantic molecule, and that's called the proteoglycan aggregate. So now it's really confusing, right? We got the class aggrecan, we got the subclass aggregate or aggrecan, and then we got the proteoglycan aggregate. It's where they can clump and mate in groups. So yeah, it gets confusing. But here's what we've been talking. This is a proteoglycan molecule right here that we've been talking. They float around, but they can become sociable. And here's a big, long molecule of hyaluronic acid. And they can link to this protein and talk about the weather and about COVID-19 and social. But notice the social distancing. They're still, they're very negatively charged, so they won't be grabbing and touching onto each other, right? But where's the proteoglycan? This is the proteoglycan right here. Where's the gag? Glycosaminoglycan is one strand right here. Chondroitin sulfate specifically. The short ones are keratin sulfate if you want to get technical. Okay, so this is a specific type of gag, right? This is an aggrecan because it only has chondroitin sulfate, keratin sulfate on it. But there's other types. But in the disc, this is what you find. Some of the new research says these guys don't like to bind as much as we thought. So these might be kind of off by themselves. They don't always hook up on hyaluronic acid. Okay, so aggregans aren't super sociable. Proteoglycan aggregans typically don't aggregate with hyaluronic acid that much like they used to think. They prefer to be alone. Therefore, there's not too many of these aggregans floating around the disc. They're more in proteoglycan aggregan form. There's not too many aggregates floating around. In other words, that's wrong. That should be aggregates in the disc. They prefer to be in proteoglycan aggregan form. See how confusing this can be? 475. Let me fix that. It should be aggregates. Okay. In vivo, though, in other tissues, like in your knee joint and in that type of articular cartilage, they form tangles, and they under the electron microscope they look like cotton wool, kind of. They're all kind of tangled up together, filled with water. Okay, they all attract and hold water because they're made of proteoglycans. All right. We looked at this again. Remember, it's the E2 domain is the trapping. That's where water is trapped mainly by the chondroitin sulfates, a couple keratin sulfates in there as well. There's an electron microscope of one. You can see it. But that's an aggregate. That's not of the disc, though. It's just of articular cartilage. There's just another picture of what it looks like. I think we looked at that before. So who cares about this water stuff? Why am I making such a big deal? And we've discussed this before. Nucleus pulposus, about 80% water. But it's really important because water is incompressible. Water, that watery nucleus is corralled within the center of the disc by the lamellae. When axial load pushes down and the water tries to squirt out, that's not going to happen, and the water becomes rock hard and acts as the ball bearing point, and that's where you pivot. There's a nice cartoon. There's axial load coming down. Water's trying to get out. It can't. The lamellae are holding it there, and that thing becomes the pivot point normally. And then all hell breaks loose, right? When you rip the back of the disc, completely destroys the biomechanics. The axial load shifts over the annular tear. It also too much of it goes back here as well, and you wear out your Z joints with the passage of time. Not everybody, though, succumb to that, but that's kind of a common, the beginning of the end, so to speak. Everything I said, it can't explode, it's corralled, it acts as a ball bearing. Okay. All right, so I think that's a good place to stop. We're going to start talking about some biomechanics, it looks like. So we will do that on Wednesday. Okay, see you later.